We are very happy to introduce the next session. The session will be held in English because we have some international guests here. And the first speaker is Didier Surdé from Zurich, and he will talk about low coverage and capture sequencing approaches in Ewing sarcoma.
Good afternoon, everyone. Thank you very much for the organizer, and thank you very much, Uta, for giving me the opportunity to present some of the results that we're conducting. So, yes, my name is Didier Surdé. I'm a group leader, I would say kind of basic researcher with also some focus on translational biology. And so this is actually a study that we started almost 10 years ago with, as an ancillary study, as a prospective ancillary study of some of the clinical trial that I'll present here. Yes, so the focus here is Ewing sarcoma. I don't need to introduce much of this sarcoma, but of course, what characterizes this sarcoma is the fat heads translocation. And as a pediatric and young adult sarcoma, we know that there are few recurrent alterations, but we and others have been able to identify some recurrent alteration, including STAG2, that is present about 20% of this tumor at the time of diagnostic, TP53 about 5%, and deletion of CDKN2A. So we don't have yet, these are all tumor suppressor, right, that are lost. So basically nothing much that we can target there, even though my group and many labs are trying to find some targeted therapy for this alteration. I'll just tell you a bit what we know at the moment, for instance, about the most recurrent secondary alteration, about STAG2. So we have been able to show that STAG2 in combination with TP53 and CDKN2A favors the oncogenic transformation of mesenchymal stem cells by E-fly. We and others have also shown that really STAG2 is a recurrent event in Ewing sarcoma, and we also recently demonstrated, and I will not go into any biological detail, but we demonstrated how STAG2 favor the metastasis of STAG2 mutated cells in Ewing sarcoma. There are also some evidence showing that upon treatment, the STAG2 mutated cells are prone to resist chemotherapy and radiation therapy, and usually at the time of relapse, they are more recurrent in the patient. So with this feature, STAG2 being associated with metastasis and with recurrence, it's not very surprising actually that if you look by what we did as a whole genome or target sequencing in Ewing sarcoma primary tumor, you can see that the patient with STAG2 mutation actually have poorer prognosis in the entire cohort, but also for localized patients. This has been now confirmed also using immunohistochemistry, including in local and metastatic cohort, and also recently published that if you look at the consequence, the transcriptomic consequence of STAG2 loss, you can identify some gene signature that again demonstrate that the loss of function of STAG2 is associated with poor prognosis, increased metastasis events. So not only we have STAG2, I'm using here as an example, but we know that the combination, for instance, of STAG2 and TP53 mutation, I don't know if there is, yes, we see that patient, it's quite rare, but that have both mutations really have a dismal overall survival. And we know that there are also some recurrent copy number alteration in Ewing sarcoma, 1q gain and 16q loss, which are also associated with poor prognosis. So can we use actually these biomarkers to basically also stratify our patient? But the question is, when you do biopsy, of course, you're only taking a part of the tumor. And you are not also sampling longitudinally this tumor. So a solution to address this question is the use of liquid biopsy. And you're probably familiar, but just for those who would be not, liquid biopsy are used for different aspects, but here we'll focus on the circulating free DNA. And basically, tumor cells or normal cells, when they die, their genomic DNA is fragmented into nucleosome size fragments around 150 to 170 base pair. And these tumor DNA fragment or circulating free DNA fragment are circulating into the bloodstream. And basically, these can now be captured, and we can nowadays investigate many of the aspects that we are interested here in Ewing sarcoma. So for instance, we can detect the translocation, we can detect the deletion and point mutation, as well as amplification or chromosome alteration. The fragmentation and epigenetic marks is also a very interesting and hot topic, and the next speaker, Eleni Tomasi, will probably tell you much more about this aspect. So I'm here the first speaker, but I would have to say that I'm not the first one really to be interested in Ewing sarcoma circulating tumor DNA. And Markus Metzler, also the first speaker here, was really a pioneer in the Ewing sarcoma circulating tumor DNA, as well as Eleni and also some group from the United States and part of the COG. But what we use here, we decided to perform low coverage whole genome sequencing, which basically allows you to capture the chromosome amplification and alteration. And we also design a target capture panel with the idea to capture EWS translocation and the most recurrent alteration. So our cohort is actually mostly patients from the Euro-Ewing 2012. It's a phase 3 randomized European trial, including Ewing sarcoma at diagnostic. We're also including combinary-free. These are the extrapulmonary metastatic patients, so very high-risk patients. This is only open in France. And then we also had the opportunity to get some sample from Recker or MAPIAC, which is the equivalent of the INFORM German study. So altogether, we have been able to profile using a low coverage and whole genome and target capture sequencing. We've performed this analysis for over 200 patients. So low coverage we mostly did from at the time of diagnostic, and the latest time point was for the target. We also use intermediate time point, but we've now really a nice collection of samples that have been profiled, and also important here that was a prospective study, so we also have all the clinical annotation, but because it's prospective, you have to be patient until the end of the treatment, the trial, right? So here is an example of what you can get from the low coverage whole genome sequencing, and basically what you see here is a typical amplification that basically reflect the presence of circulating tumor DNA. You can see a classical 1Q gain, a 16Q loss, and this patient has actually also a chromosome 5 amplification. That was at the time of the diagnostic, and actually if you analyze this at the 1 year after the end of the treatment, you can see that basically the profile is completely flat and this patient is tumor free, and therefore you cannot also detect circulating tumor DNA. Now, as mentioned, we also have deletion of CDKN2A. These are just two examples, and you can also nicely see using low coverage whole genome sequencing here deletion at the CDKN2A locus. that's part of a patient of the combiner 3, where you see again a kind of similar pattern with 1Q gain, 16Q loss, a loss of 13Q, and that was at the time of diagnostic. And actually, this patient at the end of during the maintenance retained mostly the same profile, except for maybe, if you can see here, a small focal deletion. And this focal deletion on 17Q actually contained the RAD51D, which was actually never described in Ewing sarcoma cohorts. And that could have been actually a potential therapeutic indication, because this is clearly associated with response with PARP inhibitor or ATR inhibitor. So by the time we did this analysis, it was unfortunately too late, but this also gives you some ideas where for some patients, some rare patients, we could identify some alteration where we have actionable treatment. Now for the target sequencing, here you can see that we capture basically the exon 17 to 11 of EWS-Fly. You can see that we can really enrich for this region. And now if you focus, we only capture the EWS part, but you can also, because you have spanning reads, find the exact breakpoints between EWS and Fly1 here highlighted for one patient, and these are all the breakpoints that you can identify. So this is true for EWS-Fly, but this is also true for EWS-ERG patient. Now, similarly for STAC2, we targeted all the exons of these genes, and actually quite interestingly, as I was mentioning during the introduction, we see that this patient had absolutely no STAC2 nor TP53 mutation at the time of diagnostic, and we can see that at the end of the consolidation therapy, we see that this tumor had both TP53 and STAC2 mutation. Now we can also correlate the level that we measure using the target capture sequencing for EWS-Fly, and you can see here the copy number alteration at the time of diagnostic, and in this particular patient at the time of relapse. And in addition to the capture, we were able to mention that this tumor also was carrying the TP53 STAC2 mutation. That's another pattern where you can clearly see the alteration at the beginning. You can assess circulating tumor DNA for EWS-Fly using the target sequencing at different time points, and you can see that after 2 rounds of chemotherapy, already the detection of EWS-Fly was completely absent and again remain undetectable, as well as the low coverage whole genome sequencing. So altogether, here we have now a large cohort that we provide with a low coverage whole genome sequencing, where you can see this kind of genomic alteration. We can also capture EWS X translocation and identify some of the most recurrent mutation, and we are actually actively also collaborating with Eleni Tomazu to perform this fragmentomics analysis, and want basically to combine all these metrics to find what is the best, or what are the best combination to predict presence of circulating tumor DNA, and also include some information regarding recurrent alteration. So at this time, I wish I had some more data to show you, but at the moment, we're really compiling all this data. So this is just some preliminary data where you can see that on the entire cohort, we can really measure the tumor fraction based on the low coverage whole genome sequencing at the time of diagnostic, and we can see that there's drop of circulating tumor DNA. These are just 2 different methods here to measure the tumor fraction based on low coverage genome sequencing, but you can see that the correlation between both approaches is quite good. So we have so far completed the cohort sequencing. We can detect EWS-fly translocation. We still need to complete the mutation calling for the entire cohort, and we're actively collaborating with Eleni Tomaso here to also implement the fragmentomic approach that you will hear in a minute. And basically also very important is that we have access to all the clinical information, and hopefully next time that present the data, I will be able to have a finalized story. So I would like here to thank you, and also thank the people that are involved in this project. This is a project that I started at Institut Curie with Gaël Pierron, Steli Ballet, and now continuing with Lieke Moes, a student in my lab. The bioinformatician at Curie, Victor, Camille, and Eleanor, very much involved. And of course, our collaborator Eleni Tomaso, Peter Peneder, and also at Erlangen, Markus Metzler, who also performed some digital droplet PCR of these samples, and Manuela Krumbolz. Of course, all the people involved in the trial. And last but not least, I think also the clinician here, the nurses, are really key to collect all these samples. It's a tremendous work, and of course, patient and family. Thank you very much, and happy to answer any of your questions.
Thanks a lot for this wonderful presentation. Are there questions? If not, I have a question. You said in the beginning, and this is told by a lot of liquid biopsy people, that you can find like the entire tumor heterogeneity in the liquid biopsy. Your former group in Paris showed with this paper from Francetti that like only tumor cells that have like regulate down the EWS-Fly1 would go into the bloodstream. I thought when you showed this one patient example where you found in the very end this STAC2, whether it wasn't there even before and it was just not detected in all these noise.
Yeah. So yes, thank you very much, Uta, for your question. So if I mentioned that liquid biopsy can basically average all tumors, this I would say that still has to be proven, but it's probably better than just doing a single biopsy. So, but yes. And for your question, I think so the EWS-Fly low are potentially a source of metastasis. So this would, using circulating tumor DNA, you would still detect, right? Because we're at the genomic level. And then your question is that the STAC2 is more associated to the metastasis. Now, for this particular patient, I don't know, and I don't have tumor at this time point. I don't know if the STAC2 is present at the original tumor site or if it's in the metastasis. We will expand the cohort, and hopefully for some of these patients we'll have tumor because some of them were included in the MAPI Act, where we have access to the tumor material, and will potentially be able to address your question.
Just a short question from me. In AML, we have some data that STAC2 mutation leads to an upregulated MEC signaling at its potential strong target. Do you see some changes in genetic signature in your
So we are actively looking for targeted therapies for STAC2 mutated Ewing sarcoma, and the MEC signaling is not, I would say, a hot topic at the moment, at least in Ewing sarcoma. Yes, we are hopeful for some other targets, but as always in Ewing sarcoma, this is a very heterogeneous tumor. And when you find within 2-3 entities, then the fourth one tells you, but it's not the case. So
it's entity specific.
Yeah. Thank you very much.
Next speaker will be Eleni Tomasou from Wien, and she will talk about ctDNA detection and classification of Ewing sarcoma.
Yeah, thanks also from my side for the invitation. I'm very happy to be here. Apologies, my German is not good enough, so we have to have this session in English. We will continue with liquid biopsies, and the focus is Ewing sarcoma, but what we apply in Ewing sarcoma could be easily applicable also to other sarcomas. So Ewing sarcoma is our favorite disease up to date, but of course we also started looking at other sarcomas as well. And our study started a few years ago as a proof of concept study. And our motivation was, can we follow the epigenetic state of Ewing sarcoma tumors in the blood? And Ewing sarcoma is an interesting disease because, as Didier has very nicely introduced, has very few genetic aberrations, but has a very unique epigenetic signature. So the epigenetic state of Ewing cells provides a very nice biomarker for tumor detection and monitoring and classification. So we started a few years ago asking the question, can we detect epigenetic signature in cell-free DNA? And back then, this sounded like not a very valid idea. But we thought, let's try and see what's happening. And I'm very happy to say that from a proof of concept study, this now has evolved into a study with a potential translational impact. Hopefully we can bring our assay to the clinic in the near future. So this is the aim. So cell-free DNA is fragmented, and the fragmentation patterns of the cell-free DNA carry epigenetic information. I will not go into many details because by now there are a lot of studies describing this same thing. But what you have to keep in mind is cell-free DNA that released in the bloodstream has an average size of 167 base pairs, and this is exactly the size of the DNA that is wrapped around nucleosomes. I will not go into the biology, but just this is the important information you have to keep in mind. So the fragmentation patterns of the cell-free DNA carry epigenetic footprints of the cell of origin from where this cell-free DNA fragment has been released. So looking at the fragment size distribution of the cell-free DNA, even at the end motifs of the DNA, we can start learning about the epigenetic state of the corresponding cell of origin. We can start doing nucleosome position. We can even do some kind of chip-seq on plasma to identify histone patterns. And of course, we can do DNA methylation. And all of this has already been studied and has been proven the last few years. It's a lot of research on this field. And now it's clear that by doing cell-free DNA fragmentomics, you can very nicely detect tumor DNA. You can infer the tissue of origin of the corresponding fragments. And there are some studies. One was, the original one was done in Graz by Helen Heitzer and Michael Spacher, where you can even infer gene expression by looking at the epigenetic state of promoters of genes. So this is all nice. And most of the studies so far has looked at different elements separately. We have published a few years ago a study where we looked at cell-free DNA fragmentation patterns in plasma samples from Ewing sarcoma patients, where we did whole genome sequencing and some complex machine learning tool development. I will not go into details. This is studied, and all the tools is published, and all bioinformatic tools are publicly available. But what I am very proud to say is that we develop one assay that fits all, and that we can use for confidentially detect tumor. We can classify tumor. We can say, yes, this sample comes from a Ewing sarcoma patient. We can even monitor tumor evolution, looking at the epigenetic state of tumor cells. We can quantify tumor DNA, and we can even monitor tissue damage. And this it's especially applicable for samples that have been collected during therapy. So this is all nice. Can we apply it to the clinic? It should be feasible, this assay. It requires less than 10 nanograms of cell-free DNA. Sequencing cost is constantly falling, so it's not so expensive to do it, and does not require access to tumor DNA. So we don't need to have any prior knowledge about the tumor. So this is a proof of concept study. Now we want to move a step forward and seriously make an effort to implement this to the clinic. And this diagram here I took from a nice review article published a few years ago, where it summarizes very nicely all the steps that we have to take now to bring this assay to the clinic. And we are still in step 1. So we still have to ensure the analytical validity of our assay, we have to establish the clinical validity, and to show clinical utility. And we are very happy that we got a big grant from the Vienna Science and Technology Fund, together with Uta Dirksen and Marcus, to go on and do exactly these things, to bring this assay to the clinic. And I'm very happy to say that in the meantime, we even joined forces with more people. So I'm happy that the Medical University of Graz, Bernadette and Michael Seidel joined forces. So we are also getting material now from Graz. We are discussing also to get material from the Eurovision Consortium. Hopefully this will work out in the near future. We're getting material from the UK, hopefully. Of course, the Sandana Kinderspital worked closely with us together, and DITHIA, of course, has been a very close collaboration with us along the years. So our aim is to validate our liquid biopsy-based molecular diagnostic toolkit for Ewing sarcoma, and use this as an example also for other pediatric sarcomas. So far, we have been doing whole genome sequencing for our assay, where we generate data and then we apply machine learning approaches for data integration. And so far, we have looked at cell-free DNA fragmentation patterns. We look at sequencing coverage at region of interest, nucleosome positioning, and also on copy number aberration, structural variation, which is very relevant for sarcomas. So this works very well, and we can very confidently develop machine learning classifiers for tumor detection. So we can set apart a sample that comes from a Ewing patient versus a healthy donor, and for tumor classification. So if you have plasma samples from Ewing sarcoma and other sarcomas, we can confidently say which sample comes from Ewing sarcoma. But still, we feel in order to bring this to the clinic, there's still a lot of work to be done to further optimize and increase the sensitivity and specificity of our assay. So currently, we are even developing further our bioinformatic tools and our machine learning approaches, taking into account the end motifs of cell-free DNA. So this is something we haven't done before. We are looking into this now. And we are constantly looking for any other cell-free DNA-based metrics that we could incorporate here to improve our assay. One thing we cannot do with whole genome sequencing is to look at DNA methylation patterns of cell-free DNA fragments. And DNA methylation is a very interesting epigenetic mark because it's very stable. And looking at recent publications from the GRAIL project, so this is the Circulating Cell-Free DNA Genome Atlas, where they compare different assays for tumor detection in a large cohort of adults. This is thousands of donors for early cancer detection. Their main conclusion is that DNA methylation is the best marker for detecting cell-free DNA. So in their study, this study was published last October, so it's a recent paper, and they were comparing whole genome bisulfite sequencing to whole genome sequencing, and it seems that DNA methylation seems to work a little bit better. So it's something we cannot ignore. So going forward now, we thought we should also be incorporating DNA methylation. And in the meantime, as technology goes on, an assay was developed that allows us to monitor at the same time, in the same plasma, cell-free DNA fragmentation patterns and DNA methylation. So it's this enzymatic DNA methylation-seq assay that, I don't know if you have done bisulfite sequencing in the past, but doing bisulfite sequencing for DNA methylation is a little bit harsh to the genome, so it destroys it. So it cannot be used, bisulfite sequencing cannot be used for detecting, for keeping fragment size. But now with this assay, we do DNA methylation profiling using an enzymatic approach, which preserves fragmentation patterns. So by doing EM-seq, we can combine fragmentation analysis with DNA methylation information. And this is now where we are standing, where we have generated now a lot of data, and now our difficult task, or the major task of Peter and Nicholas from my group, is to manage to integrate this data by performing some neural network analysis and machine learning. Unfortunately, I don't have the results yet from here, because to confidently show results here, we will have to have a sufficient number of samples. But just I want to very briefly touch what we want to do with this approach. So this is just a figure summarizing a treatment therapy, the therapy protocol for Ewing sarcoma patients. So we want to develop our assay that can be used, for example, at the very beginning, at the initial diagnosis, when someone comes in the clinic and there is the suspicion that this patient might have Ewing sarcoma. So we want to have an assay for this. We want to have an assay that we can monitor patients during therapy or after therapy for detection of relapse, and also for tumor evolution. And at the moment, we are focusing mostly at mode 1, initial diagnosis at baseline. And we want to optimize and validate our assay as a complementary minimal invasive assay that we could offer in patients at the very beginning, together with tissue biopsies or MRI and PET scan. Let's say you have a child and comes in with some pain. So instead of having to wait for biopsy or to organize a PET scan, you can just take a blood, and then we can do our assay and then confidently say very quickly if this patient has tumor, and if yes, is it Ewing sarcoma? And hopefully this way we will also contribute to earlier diagnosis. Then, of course, we can ask, does this patient has metastatic or localized disease? We can infer this from our epigenetic patterns. And also, it will be very interesting to see for patients that were initially presented with localized disease, but later on develop relapse and metastasis, could we already see at the very beginning acute metastasis? And this is a very difficult task, but we are trying to address it with some complex machine learning algorithms. Also, can we predict therapy to relapse from the very beginning? And can we predict which of the patients would develop relapse or metastasis later on? So these are now the aspects we are trying to work on. And to answer all this, we need a proper cohort, a very nice cohort. So based on our power calculations, we need at least 200 patients, and they have to be equally distributed. So we want to have at least 100 patients that were diagnosed with metastatic disease at initial diagnosis, and 100% with localized disease, and then again, half of them should relapse or not relapse later on. And importantly, we need controls, so we need at least 100 healthy controls. We also want to have as controls plasma cell-free DNA from patients with other sarcomas, and also we want to make sure that whatever we do, we don't only discriminate healthy from something else, but we really detect cancer. So we also want to include other non-cancerous syndromes that could be relevant for our analysis, most importantly, inflammatory cases. And so far, so this is ongoing, and so far we are we have 50%, almost 50% of what we would like to achieve by the end. So we have already processed 91 Ewing sarcoma plasma samples collected before therapy. We have generated data for 62 healthy controls. We have 3 samples from non-cancer conditions and 12 other sarcomas, and this keeps, we're increasing the number, I hope. By the end, within the next few months, I hope we reach this number so we can confidently develop our machine learning classifiers. And with this, I would like to thank a lot of people. So working with pediatric cancers and sarcomas, it's a multi-center effort. So of course, a big thank you goes to Uta and Marcus. A special thanks goes to Didier and all of the collaborators that have contributed samples today. And I hope we can continue collaborating. And if you feel you can contribute to this study, please talk to us. And if I have one more minute, I want to make an announcement. So we'll be very happy to meet with you again in Vienna in May. So we are organizing the EU SARC meeting this year. So it will take place in 18th to 20th of May. We have a keynote speaker, Kim Stackmeyer, will be joining us from the US, and there are a few other good guest speakers with exciting talks. Talking about liquid biopsies, I'm very happy that Isidro Cortes-Quiriano will be joining us. He does sarcoma study of liquid biopsies using long-read sequencing nanopores. I'm very curious to hear more what he will be doing. And last but not least, this is a photo of us in Vienna. We are very happy because recently we got an ERC consolidator grant dedicated to pediatric sarcoma, so we are hiring. So if you are interested in sarcoma, pediatric sarcoma research, or you know any young scientists or clinicians that they want to join us, please spread the word. Thanks a lot.
Thank you very much for a nice talk. Are there any questions? If not,
I may, because Uta asked earlier regarding if with the liquid biopsy we capture all tumor cells. This was what I thought when we were starting. But for our study, we got much tumor DNA from DTA, actually, from the published study, where we were comparing copy number aberrations inferred from liquid biopsy or from the matched tumor. And in many cases, they match, so this is nice. In some cases, you see more copy number aberrations in the plasma, but in some cases, you see less. To me, this implies that not all tumor cells release cell-free DNA with the same efficiency. And to understand this better, we have to still compare. We don't know enough, I think. So we will have to be able to answer this, we should systematically compare liquid biopsies to matched tumors.
Yeah, another question would be, but this is really difficult to do and difficult ethically wise and so on, is maybe also the time point. Because Marcus has shown that the cell-free DNA there's another peak like after the first course of chemotherapy, and maybe we would get the best results if we would take like every chemo day a sample.
Or it would be nice to get like 24 hours after therapy, 48 hours after therapy, yes.
One question, Peter?
Do you have information on tumor heterogeneity? We know from completely different cancer types like colorectal that there is a difference between primary tumor lymph node metastases and liver metastases. Obviously, in a liquid biopsy, we would hopefully see all three different patterns. Do you have experience with something like that, or evolving changes over time?
So we try to look at copy number aberrations, how they change over time. For the cohort we had, it was not very systematic, but still we had a sufficient number of samples, but we could not come up with, at least for our samples, there was not something systematic to say, oh, chromosome 8 amplification increases. It was rather, so we couldn't make any conclusions. We tried to look, but only looking at whole genome sequencing data, liquid biopsies, to look at epigenetic state, how it changes over time. We had some indication that the state of epigenetic state of STAC1, which is very relevant because of STAC2, it becomes more active over time, but it's very preliminary, so we will have to, let's say, to confirm this. What ideally would be like to be able to see if we can do in the future, now that we all generate single-cell data from tumors and we know the different cells that make up the tumor microenvironment, or even if we have transcriptional heterogeneity, if we can see these signatures in the blood. And this is something hopefully we will be interested also working on, but we don't know yet if we can monitor this in the blood.
Thank you. If no other questions, we will continue. Thank you very much.
Thank you.
Now we will continue with the next talk from Markus Metzler from Erlangen, and it will speak about the impact of radiation on detection of ctDNA.
Thank you very much. You might wonder why radiation and circulating tumor DNA. And well, one answer could be after Didier and Eleni, there's not much to say anymore. But that's not really the reason. But puzzles me already for quite a few years is the question, what exactly does the level of ctDNA tell us? So we don't have this direct correlation between treatment and imaging and this blood sampling. And this obviously cannot be answered in the patient straight away, and we need a kind of model for this, and this is what my talk is about. So the motivation is, I will talk about shortly about the motivation, then what we found out in the Ewing 2008 trial, the EFFECT study, which dealt with the circulating tumor DNA in Ewing sarcoma. And then I will show some experimental data on radiation and ctDNA, and then at the end, the objectives of the upcoming or following EFFECT 2 study. As I said, the motivation was first generally, there's quite a few, there's quite some data on the effect of surgery and chemotherapy or targeted therapy on the release of circulating tumor DNA, but not that much on the effect of radiation. So it's less well studied. Then clinically, we do not have this correlation between ctDNA imaging and histology, at least not in a timely manner. And this is particularly in high-risk tumors because they are usually not resected, and then we do not have the tissue to compare our blood samples with. And then finally, a rather practical issue is that if you want to study this in an animal model, radiation dose is far better tunable than a preclinical model than surgery or chemotherapy in a small animal. This has been shown by Uta before. This is the backbone of the EFFECT trial, the Ewing 2008 trial. And at this time, we knew from first for some pilot patients that the circulating tumor DNA decreases rather early under treatment, and therefore we focused in this part only monitoring ctDNA during the induction therapy, which was these Wiedeblocks at this time. And to study not only single cases, but larger cohort, we analyzed 102 EFFECT patients where we took blood samples before and after the first Wiedeblock, and then before the third and the sixth, and after surgery if it was performed. For our purpose, we took digital droplet PCR and used the fusion site as genomic marker for the circulating tumor DNA because of sensitivity reasons. And then combined or compared these results with tumor volume, with risk factors, and obviously survival parameters. I'd like to acknowledge Manuela, who did a lot of the technical work there. And I pick out two interesting results from this study. First was that we saw that circulating tumor DNA copy numbers do positively correlate with the known adverse risk factors like pelvic localization, large tumors, the presence of metastasis, but somehow inverse correlation with the histological good or poor response. And even before treatment, the copy number was correlated with the, in this case, event-free survival, even in metastatic disease. So it does has some predictive value. And the thing is, which I don't show here, is that it's not just a representation of the size of the tumor, but seems to have some intrinsic feature. And this is what was mentioned before by someone, that there are obviously tumors which release more and others less, although they have maybe the same size or same localization. And this is exactly what we wanted to study in more detail. And secondly, we obviously did have patients which relapsed, and in these relapse cases, we saw that radiation had a relevant impact on the further course, and we obviously cannot dissect this clinically from which treatment element is responsible, whether it's chemotherapy or radiation or a combination of both. And this is what we wanted to mimic in a mouse model, xenotransplant model, where we somehow wanted to introduce a tumor which grows, is being monitored, then it's treated, but to an extent that a relapse occurs in a reasonable amount of time, so we can study both phases. And you can imagine that implanting tumors intrafemoral in mice and do radiotherapy on a clinical device in these cases is not that easy, but eventually Johanna and Benjamin found out how to produce consistent results. And then for another reason, we needed to mimic somehow tumors which represent the radiosensitive tumors and the resistant tumors. And therefore, we tested first in vitro different Ewing sarcoma cell lines, and to identify the proper dosage of radiation and the proper radiation scheme. And we tended to use the same fractioned radiation as we do in patients. And this gave us the opportunity now to combine the blood sampling and analysis from the liquid biopsy with small animal imaging as close to the clinical situation as possible. And this is the novelty with direct histological correlation, because we can take samples at any time point. And this was done by the help of the colleagues from nuclear medicine, from radiology and pathology. And to show that this didn't work only in theory, but also in reality, we see one example here of a mouse, control mouse, which has not been treated, so tumor grows. But if it's irradiated here, it goes into remission and then relapses. If cells are radiosensitive, if they're radio-resistant, they are just halted for a while, but then grow back, and some remain in remission. And we've been using this model then to study certain aspects. I'll show you in a minute. The first aspect is that there is a correlation between copy numbers of ctDNA and tumor volume, but only in tumors which continue to grow, those are chemo-resistant, the radiation-resistant, but not in the sensitive cases. But there's not a clear pattern there, so there's some heterogeneity. It's not just that they react all the same, but we have to find out what's the difference there, why this is such a heterogeneous behavior. Then secondly, we've been using a series of immunohistochemistry to study, the tumor content, the proliferation, the mitotic activity, and the apoptotic activity within the tumor at certain time points to exactly study the effect on the therapeutic intervention in parallel to what's going on in the tissue and what's going on in the blood. And one interesting aspect that we could see for the first time, that there is kind of response early after radiation, so that cells seems to grow up as long as they can, and then eventually they die. And caspase happens early, but is kind of repaired already shortly after radiation. And we can do this for different time points, obviously. And turned out that not the necrotic area, not the mitotic activity, not proliferation correlates with the ctDNA copy numbers, but it's our model only apoptosis. And the last interesting aspect is that the microenvironment changed obviously significantly after radiotherapy. So this is here shown for CD70. It's kind of immune marker, and this is which is sometimes, for example, in this radio-resistant tumors, hardly present, but then has a very different pattern after radiotherapy, and we want to explore whether this makes tumors more accessible to immune intervention. So obviously this has been all studied in the context of Ewing 2008. Unfortunately, Uta changed the chemo backbone of the current trial, so we have to re-evaluate this obviously now within the new Ewing trial, but this gives us the opportunity to study extra aspects, and particularly the effect of radiation and the long-term follow-up, where we expect to be able to pick up at relapses at an early time point. And this gives us the opportunity to study many aspects which Eleni mentioned just in the previous talk. And finally, as mentioned several times, the basis for that is that we do have these samples, and I'd like to take the opportunity to advertise to contribute to this trial. And what confuses people most was that there are similar looking schemes. One goes to Essen and one goes to Erlangen. And yes, this is right. This is meant to be like this because we do have to handle the samples slightly different, and we are in a close connection with each other, but it's important that the samples are collected in the proper way, and this is all shown in very easy to handle flow charts. And if not, Sabine Semper and Frau Jabbar are always there for questions, and all other questions, please to me. Thank you for your attention.
Thank you for your talk. Are there any questions? Perhaps I may start with one. What would the clinical meaning? You try to detect patients which are resistant or progressive under radiation treatment. That's the most important point, or do you see some other aspects?
We use radiation as like well-tunable therapeutic intervention. We want to know the timely correlation between your therapeutic intervention and the response to that. We do know, obviously, from surgery that immediately after surgery, ctDNA levels go up and then they go down. But this is not the situation in Ewing sarcoma or any other sarcoma. For chemotherapy, we know the typical behavior, but as Uta mentioned before, we do see some cases where it goes up after chemotherapy, and others, they seem to be rather resistant to that. And if this could be an early indication which are chemo sensitive or not, you could maybe earlier stratify them.
Okay.
And therefore, we need a very specific intervention, and polychemotherapy in mouse is a complicated issue.
Thank you. If no other questions, thank you.
Okay.
Okay, so I'm happy to announce the final speaker for the session, and his talk will smoothly lead to the next session because Maxim Kebenko will talk about liquid biopsies in GIST.
Just have, okay. I'm going to present a short update about our study. We want to know or to explore a predictive value of liquid biopsy by patients with high-risk GISTs. And why do we do it? Because liquid biopsy is technically not easy, but it's easy to take samples. We need blood samples. We need, of course, detection of the driver mutations on the tumor tissue, and we can detect the mutations in the blood. We can measure the dynamic of ctDNA, and here on the right side you see a clinical case where you see that the patient with the treatment with sunitinib, it shows he shows a decrease of ctDNA before you see the clinical response in your imaging, and on the other side, before the clinical relapse, you can see the increasing values of ctDNA. So it could be important for early diagnosis and treatment management. And we can, of course, the time of different TKE treatment options, we can detect secondary mutations which that lead to resistance. Here, publication from this year, for example, here the case of a patient with a treatment with regorafenib and developing secondary mutation in exon 17, and then you see that this clone is progressive 100 of the days later. So we can do this, say, in our lab in Lübeck. It's established, and it's one of the points of our project. We include patients with the both typical driver mutations, detected first, of course, on the tumor tissue. We don't need the mutation state to include the patients. We can include every patient with a diagnosis of GIST, and we can then in the screening time wait for the end diagnosis, molecular diagnostic, and then if the patient doesn't have any of these both mutations, we can exclude them. It's no problem. We include patients with localized or localized advanced disease, but also patients with oligometastatic disease who can be cured by surgery and TKI, and of course, patients with a stable clinical state to be treated. Our study design, we plan to screen 100 patients, and we expect something like 80% of the patients with positive ctDNA values before treatment, and something about 20% which who are negative. We will monitor the ctDNA in positive patients at the beginning of treatment. If neoadjuvant or not, it's not so important. Patients may be treated neoadjuvant if it's indicated, and we of course do it after resection during our follow-up. And the patients who are negative for ctDNA, they will get only clinical follow-up. Our primary endpoint is detection of positive ctDNA tests after resection, with or without induction treatment. And of course, in our secondary points, we want to know more or to learn more about specificity and sensitivity. We don't know really or don't have really available data about regarding these both parameters. And of course, we have some translational projects, proteomic analysis, and so far secondary endpoints in our study. Our current state is we have 4 centers which are initiated, and we have 2 patients in Lübeck. Planned centers are Dresden, Hannover, München, Essen. I hope the initiation process works now, or better, or faster. And 1 youth center is Erlangen. And the next initiations will be in Göttingen and Erlangen. And I think it was the last slide. It was short, but it's only a short update of our study.
Okay. Thanks a lot. Are there questions from the GIST people, maybe? Eva.
Eva Wadelmann, German or English? What do you want? You can get what you want.
What you prefer.
Okay, so English. I'm always wondering about the question how we can decide which tumors shed DNA into the plasma and which don't. Because, in fact, in the solid tumors, this might be a problem. So what do you do with negative results? I mean, you said what you do in the study, but what do you expect? Do you think that all the people having no DNA in the blood have no metastatic disease, and all the others which do have metastatic disease? I mean, is this the theory? Do you think this is true? Or
I think it's too simple to say it in this way, because we don't know about part of the patients which are initially negative, what could be during the follow-up after treatment, and the study will explore patients in the curable setting. About metastatic situations, not oligometastatic excluding, we don't know anything. So I think these questions will perhaps be answered during the study, if we have some follow-ups.
I think in Ewing sarcoma we found it in every patient unless there was surgery in the beginning, right? Yeah.
Yeah, but I think the biology of Ewing sarcoma and a solid tumor like this is so different. I mean, always if I look in these bone marrow smears looking for tumor cells, I feel like it's incredible that there are swimming tumor cells in the bone marrow, which never happens in GIST, for example. So I would expect the biology to be completely different. And this is why I'm wondering how safe the approach will be in the future. Because what we were just kidding around was that one day pathologists will not have to do anything anymore, which is good, of course, because everything is done by MRI and blood throwing, and then just some NGS or fragmentomics or whatever. No, I'm kidding, but the question is, of course, is this in every tumor entity an appropriate approach, and would you give an aggressive therapy to a patient because of a liquid finding? I mean, and just it's not so difficult because you give imatinib or whatever of the other lines, but in a sarcoma?
I have shown it on one of the first slides. The really important question is what's about specificity and sensitivity of our assay? Because usual digital PCR is not really sensitive in GIST. You have a specificity of 90% and more, but sensitivity only 40-50%. And we don't know anything about it by using the digital droplet PCR. And I think then we can say if it's really sure approach, stable approach to predict relapses and to stratify treatment options. But at this time point, it cannot give you any answer if it's stable enough to use it in the clinic.
Danke.
Okay, thanks a lot. So I would like to conclude this session with maybe 1 or 2 sentences. We designed this session because amongst the people who work on the program of this whole conference, there was the idea that there's a lot of ongoing in liquid biopsies, but not everybody had a clue what you can do with this and how is the technique. And I think all these talks showed that there is a lot ongoing. There are a lot of different techniques you can use. Part of this is still kind of a moving target, and at least the Ewing community showed that it's also about labs working closely together to come to further conclusions and to make something meaningful for the clinic out of all this. And I think that's especially in the sarcoma the case.